Hello everyone, myself Pankaj Kumar Burwa from Nagaon district, Assam. So my project was the estimation of oxalate ion present in different types of colocasia and the comparative study of etching capacity in them. So what we have done, colocasia is a common plant which is available to our locality. So since I belongs to Assam, so Assam is full of colocasia, it is there. In my school campus also it is colocasias are there. So what we have done, we collected 5 different types of colocasias from our locality with the help of our students. Then we tried to find out their oxalate ions present in them. So what we did, we collected the colocasias wild colocasias and tried to find out the oxalate ion with the help of some titration which is there in the class 12 syllabus. So in class 12, the students has to study the oxidation reduction method. This titration is also there in the chemistry practicals. So some investigative projects are also there in their syllabus. So what was my approach is that, so to bring the syllabus from the out of the classroom to the nature, so I asked the students to find out different types of colocasias which are wild, which are not actually cultivated, so which are available to our locality. So they collected 5 different samples. Then we tried to find out the oxalate ions. So oxalate ion is the main cause who causes itching in our throats. So when we think that if the colocasias can be removed from the oxalate ion can be removed from the colocasias, then these wild varieties also can be made edible. So our one basic purpose was that to remove the oxalate ions from the colocasias and make them edible. So in doing this, so what we can do, the poor people though who cannot afford too much of colocasia which are cultivated, so they can use the wild varieties. So one other aspect of our project was that to make these wild varieties available to the local people or make them edible. So other aspect is that, so make some snacks or some powders like that from where we can get some different food products. What our students did, so we collected the colocasias, the roots usually, then we take 50 gram of the sample, then we remove the peels, then it is soaked nicely, then it is boiled with dilute sulfuric acid for 10 minutes, then the solution was collected, the extract was, the volume was made up to 100 ml. Then this volume was titrated against 1/20 normal potassium permanganate solution. So then it is seen that different types of colocasias having different oxalate ion concentrations. So this is the methodology what we have used. So potassium permanganate solution is prepared by 1.58 gram of potassium permanganate dissolved in 1 liter of solution. That is how we made the solution. Then with the help of this potassium permanganate, we titrated the oxalate ion concentration present in the different types of colocasias. So after that, what we have done, that colocasias are treated with lemon juice. So then again the same procedure is done. So again the colocasia, the oxalate ion concentrations were estimated. Then it is digested with lime water. Then again it is the oxalate ion concentration were estimated. So it is found that when the normal colocasias are treated with potassium permanganate solution, how much oxalate ion concentration was there, but when it is treated with lime water or in lemon juice, the oxalate ion concentration reduces. That means reduction of oxalate ion concentration can reduces the itching which is caused in our throat. So that means by these methods we can tell the people that these wild colocasias also can be made edible by these processes. So some experimental results are there in the next slides. So this is the data collection what we have done, the colocasia extract ordinary one. I have used this formula from chemistry. So the normality was calculated. So like this sample are there, Dutkosu it is Xanthosoma sagitifolium, this is one. Another one is Bogakosu, it is Alocasia formicata. Next it is Olkosu Amorphofolius peonifolius. It is Borkosu, Alocasia mycorrhizos, LG dome. Then next is, so then lemon juice treatment, what we have seen for the all 5 samples, that concentration is reducing. So you can see from the slides, it is reducing. So these are the experiments what we have done in our laboratories. So for 5 samples we have tested. Then this is for Bohr Kosu, this is Alocasia mycorrhizas, what concentration we have got, 0.1525 normal. So it is the results what we have got from the lime water treatment. So lime water treatment what we have got for Kola Kosu, which is called as Colocasia esculenta L Scott. This is the concentration was found 0.0525 normal. So it is for Dus Kosu it is 0.05 normal. It is for Boga Kosu it is 0.525 normal. Then it is Ol Kosu, this is 0.035 normal. Then it is Bohr Kosu 0.1 normal. So if I analyze the data, then we get a graph like this. That means in this graph it is very clear that Dus Kosu contains, that means Dus Kosu means the Xanthosoma sagitifolium contains the less amount of oxalate ion, that means which causes least itching, and Bohr Kosu contains the maximum concentration, that means it will cause more itching. So while that Colocasia was treated with lime, it is lemon juice, so that concentration were like that, then the graphical representation is like this. So that means if we see compare the values, then it is reducing. So but the same thing is there, the Dus Kosu contains the least amount of oxalate, while that Bohr Kosu contains the maximum amount of oxalate. It is the result what we have done with the lime water. So again if I see the graph, so if I do a comparative study, next sir. So this is a comparative study of the different types of Colocasias, 5 different types, then these are the results. Then if I compare this, next graphical representation will come. So from the graph it is seen that there is a variation, that means the green graph which is given the ordinary extract, then the pink one is for lemon juice treatment, the last one is for lime water. So it is clear, that means while we treat the Colocasia with different types of solution like lemon and lime, so the oxalate ion concentration gradually reduces. So there is a problem what we have got in that graph, that means in the first part of the graph, so we got some different readings. So it may be because of that this project was done carried out for months long, and because of that we have started in August and finished in December. So during this period there is a climatic condition change, and because of that some oxalate ion concentration may also differ. So from this experiments what we can say, so Dus Kosu contains the least amount of oxalate ion concentration, Bohr Kosu contains maximum amount of oxalate ion concentration, and from which we can say that Bohr Kosu itches more while Dus Kosu itches less. So by knowing these results, so I can say the people, common people, that this can be done by treating with the Colocasia with some other organic material like lemon juice or some inorganic solution like lime. So this Colocasia oxalate ion can be reduces. So this is also helpful in teaching, because these students will know how to use the experimental part which is there in the laboratory. They can link the laboratory with the nature. So finding the nature, what is there in the nature, so there is a link between the laboratory as well as with the nature. So that is why it is having a very educational significance also. So another aspect was that we have to study the microscopic observation of these different types of Colocasias which are found. This was done by our students. So this is a stem, cross section of a stem of Colocasia. So this aspect is that biology students also can understand this. That means this project correlates biology as well as chemistry. So it is a interdisciplinary project. So this project can be a investigatory project for the class 12 board exam students. So this is another aspect. So many things I have to do, but due to the limitation of time, so this much is done. So next time I am thinking to do more. So thank you.